PCR as an alternative for cDNA cloning. Like the genomic DNA, where we can amplify a specific target gene if we have a specific primers. So like that, we can also amplify a specific messenger RNA by using a specific primers. So it is also an appropriate technique for cDNA cloning. RT-PCR, it is basically a reverse transcription polymerase chain reaction, which is commonly called as RT-PCR. And this PCR has been used to quantify the expression of the RNA. So if appropriate primers they are being provided, then they can amplify the target messenger RNA and convert it into cDNA. So it is a very appropriate and good technique for the detection, quantification and cloning of cDNA. And here for in vitro reaction, we can provide first primer that can join at 3' end, if you remember, that will direct the synthesis of first strand, and then after the synthesis of first strand, then second primer at 5' end it is provided that will direct the synthesis of the second strand of cDNA, and then it is ready for cloning. RT-PCR, it is having high sensitivity, so that if you will provide a population of the RNA. Population of the RNA means that it contains transfer RNA and the ribosomal RNA in addition to polyadenylated messenger RNA. So it can amplify messenger RNA and convert it into cDNA with high specificity. So this is one of the advantage. Although it is advantageous to clone to amplify cDNA, but its screening it is relatively laborious. Another problem is that when we attempt to amplify the cDNA, if the stretch of the messenger RNA is very long, then it is not necessary that all the sequences of the messenger RNA they may be cloned. Sometimes only a portion of the messenger RNA may be cloned that will not represent the whole messenger RNA. Another drawback of RT-PCR is that the reverse transcriptase or polymerases that are used for amplification, they are error prone. Error prone means that some mistakes may be there during the process of amplification, और इसकी वजह से जो हमारे पास cDNA आएगा तो उसमें म्यूटेशंस हो सकती हैं. तो इस तरह की चंद एक प्रॉब्लम्स इसमें होती हैं. And finally, another problem may be that when we attempt the extraction of the messenger RNA from a specific tissue type, then genomic DNA may also be extracted. दूसरे लफ्ज़ों में आप ये कह सकते हैं कि एक्सट्रैक्शन प्रोसेस के दौरान मैसेंजर आरएनए के साथ genomic DNA आता है and it may represents the contaminants. So messenger RNA it is contaminated with genomic DNA. So under such situation, genomic DNA it may also be amplified along with cDNA that may be problematic. Now, because of this reason, it is desirable if we use primers that can bind at different exons so that they can amplify messenger RNA in different fragments. And on the other hand, the genomic DNA it is amplified in large fragment that can be distinguishable from smaller fragment of cDNA. And if this problem it still persists, then the sample after extraction it can be treated with DNase. Now this DNase it can specifically degrade the DNA and we have the sample of the messenger RNA that can be used for the formation of cDNA by using RT-PCR, and that is now ready for cloning. So this is one of the advantage of RT-PCR, that we can also clone cDNA by using this technique.